Good morning students, welcome to YouTube channel. So now let me discuss one of the important concept in this video, that is the search for the genetic material. Search for genetic material means, so at present we are not searching. So in 1868, almost in 19th century, mid of the 19th century we can say. So many scientists were worked on that and they have given many experiments for our study. So that is, as you know, the nuclein, one of the important substance, nuclein, which is observed or discovered by Frederick Miescher in 1868. In 1868, okay, and almost in similar time, that is principles of inheritance. Principles of inheritance you have studied in previous chapter, that is proposed by, the principles of inheritance was proposed by John Gregor Mendel. John Gregor Mendel. So almost similar time we can say 1856, it is 1856 to 63 he was conducted, okay. So on pea plant that we have studied. So now, so why I am taking this one, these 2 points here, because you have studied previously, that is nuclein was discovered by Miescher and principles of inheritance was proposed by Mendel, okay. So now the genetic material, during that time Mendel was called it as a factor, something is transferring some characters, means the characters are transferred from parents to offsprings, but he did not explain what exactly the material, molecules, which molecule is causing. He was used simply that was a factor, okay. So now Miescher was identified nuclein, but he is also not explained this is the genetic material, okay. So for the search of this genetic material, many scientists were gave their hypothecation as well as experimental proofs, okay. Let us be discuss in this video about the what you call search for the genetic material. See, the mechanism for genetic inheritance had reached the molecular level in 1926. Means almost in it's a 20th century. So previously in 18th century was started explaining about genetics, but the molecular level is reached. The search of genetic material were reached the molecular level. Will you understand? The chemistry of the genetic material were reached in almost 20th century we can say, okay. So that is 1926. Let us see the previous discoveries before this one. So Gregor Mendel, Walter Sutton and Thomas Hunt Morgan and many scientists were helped the search of genetic material, that what we call the previous discoveries by these scientists narrowed the search to the chromosomes located in the nucleus of most of the cells. Understood? See, chromosomes are carrying the informations, or that means characters from parents to offsprings through gametes, okay, from both parents. So the characters are coming to the offspring by the gametes during reproduction, as you know, okay. See, these scientists, after that the actual molecule of a genetic material remains a question, okay. So even after the search of this what you call nuclein as well as principles of inheritance, whatever factor, gene, whatever they call, no. So but after long time also it is remains as a question mark, that is what is the molecule, what is that, whether it is a protein or it is a RNA, DNA, which molecule, either it is a polysaccharide or whatever, which molecule is causing the this inheritance. Okay. So, here some experiments we are going to discuss in this class, that is transforming principle. So, this is the also called a Griffiths experiment, because Frederick Griffith in 1928, he explained and he proposed this principle, that is transforming principle, by conducting a series of experiment. He was conducted a series of experiments. So, by that he is going to conclude the what is the genetic material, okay, which is the genetic material. See, conducted a series of experiments with a bacteria. He was choosed a bacteria that is called Streptococcus pneumoniae. The name of that bacteria is Streptococcus pneumoniae. This bacteria, I hope nowadays it is more popular, because along with corona, the Streptococcus pneumoniae is one of the disease causing bacteria, which is a pathogenic bacteria, that is that cause a disease called pneumonia, which cause a disease called pneumonia. Pneumonia is the name of lung disorder, which is caused by this a bacteria, that is Streptococcus pneumoniae. Okay. So, based on this, means by using this bacteria, the Frederick Griffith in 1928 conducted a series of experiments. He observed that transformation means physical change. He observed in two kinds of bacteria. There are two strains. Same Streptococcus pneumoniae has two strains of bacteria, that is physical nature is physical forms are different. One is what you call smooth, and another one is rough. Smooth strain and rough strain. So, it is referred as simply S, and it was referred as R. So, S strain and R strain he was used, and he observed the two kinds of bacteria here, that is both Streptococcus pneumoniae. One is virulent here, another one is what you called non-virulent. One is responsible causing the pneumonia, that is S variant, that is what smooth strained bacteria. So, that causes the pneumonia, but the R strain does not cause the pneumonia. Okay, let us see how he was conducted that experiment. Okay, let me discuss now. See here, students, see here, Griffiths experiment, transforming principle. So, Griffith, either the question may ask you explain Griffiths experiment or transforming principle. Understood? See here, what he conducted a series of experiment. Just let us see. There are two strains of bacteria he was used. So, he was cultured the bacteria, that is bacteria grown on culture plate. Culture plate means he was used the culture medium for growing the bacteria. Okay. So, there are two kinds of bacteria in a same, that what we call Streptococcus pneumoniae. Okay. So, that bacteria has two colonies. He observed in that culture plate two kinds of bacteria of that same Streptococcus pneumoniae. See, this bacteria has two colonies now, they are named here smooth shiny colonies. First one, smooth shiny colonies, that is S strain, he called it as S strain. Smooth shiny colonies he called S strain. Okay, S strain bacteria, and another one is rough colonies. Rough colonies he referred it as R strain. Rough R strain. Okay. See, so how do we differentiate these two? On the physical changes only we can observe. So, there was a smooth shiny, means because there is they have what you call a mucus coat is present on this. The surface of the bacteria shows mucus coat, which is usually synthesis of polysaccharide. What you call a polysaccharide. Polysaccharide was produced here, synthesized on the bacterial coat. So, that forms a mucus coat. Polysaccharide mucus coat is observed on the smooth shiny colonies of bacteria. That is what S strain bacteria. Okay, S strain pneumococcus bacteria. Okay. Next, so this rough colonies were not showing any mucus coat on that. Okay. So, no mucus coat of that polysaccharide. So, that in this type of bacteria polysaccharide is not synthesized, hence there is no coat, hence it appears like a rough surface. Okay. So, these are smooth shiny referred as S strain, these are rough colonies. Here we used the term that is R strain. Okay. So, this you should remember first. Next, the S strain means smooth shiny colony bacteria are able to cause a disease which is called virulent. Okay. It's toxic, which is a toxic for other organisms, that is usually mice. He was used for his experiment. There are two organisms was used by Griffith, that is what we call, one is this bacteria, another one is mice. Okay. Mice. Okay. Mice means here rat we can call. Okay. So, actually this virulent S strain is a virulent, means toxic and pathogenic, either you can call it as pathogenic or virulent. Okay. So, or it can able to kill or cause a disease. Okay. So, that disease is called pneumonia. Pneumonia, the same bacteria today. So, along with corona we may observe that due to pneumonia also the what you call the same symptoms which we observed for corona as well as pneumonia. Okay. So, you can see pneumonia is also one of the disease which is caused by even some viruses are also there and this bacteria. Griffith was used this pathogenic bacteria, that is which causing a disease in a mice, that is called, the disease is called pneumonia. Okay. The name of bacteria is pneumoniae. What you call? Streptococcus pneumoniae. Streptococcus pneumoniae. Okay. Even commonly called pneumococcus. Okay. Like we use the term. See, this is rough surface, non-virulent, non-toxic and does not cause a disease. The disease is not caused here, but this one S strain caused the disease and rough strain R strain does not cause a disease. This is the main hint for this experiment. You should remember, S strain cause a disease, R strain does not cause a disease, even it is infected. When it is infected, how it is infected? So, Griffith were collected this bacteria into the syringe and injected to the mice, okay, on practical animal, that is. Okay. So, on that animal he injected and that animal whether suffered from pneumonia or not. So, he was used this series of simple experiments. Let us we see that experiments are like this here. First experiment we can consider. So, in single line I have explained here. So, S strain. See, S strain means this one, virulent, which is a virulent, okay, which is injected. He was used the injection. So, injected into mice, a healthy mice. He was cultured, that animals are also cultured mice, different mice was used. So, he used different mice here. So, that is S strain means virulent, which caused the disease. You remember, this was injected directly into the mice. Okay, that mice is died. Yes, the mice is died. First experiment is over. Mice is died. He was noted this one and he also noted it. Okay. Next, R strain was used. First experiment with S strain, second experiment with R strain. Simple experiments. Okay. So, R strain was collected and injected into mice. Another mice, because it is died, no. He was used another mice in the second experiment, injected into mice. That mice is remains alive. It can live. Okay. Mice is even after injection also even after infection of this bacteria, R strain bacteria does not died. Mice is not died at all. Okay, it remains live. Means understood no this point. It's virulent means toxic to that cause disease in this. By that disease mice is died. Understood? He observed that. Okay, in one line we are explaining, but he was used much time. Okay, so next in third experiment, what he did here, some changes he made. That is what he killed the bacteria of S strain bacteria were killed by heating. He applying the heat. Okay, he collected that bacteria and heat, after heating they were going to die. Okay, so that remnant thing is, that is S strain, heat killed S strain. Will you understand? Heat killed S strain means heat killed S strain. Heat killed S strain is injected. It was injected to another mice. That is that mice remains live. Okay, still it may alive after the infection of heat killed, because when he directly killed in first experiment, that was died. The mice was died, but after heat killing the mice remains alive. Understood? So mice were not died, because this virulent is yes, what happens here after killing, after destroying, so what happens it is, so it is become non-virulent. We can consider, no? So it's become non-virulent when it is killed. Okay, so killed means killed part, that thing is, the bacteria is injected to the mice is remains alive. Okay, next in fourth experiment he was used both strains. Which strains? Not alive, both are not alive. One is dead, another one is alive. Which one is dead here? Very important. The virulent was, he was what you call the S strain is a virulent. It can definitely kill the mice, but heat killed is not allowed to die. Okay, so heat killed S strain is also when he injected alone itself. Okay, so only if he injected heat killed S strain, mice is live. Okay, mice can allow to survive. Okay, but which is survived there, but here heat killed S strain, heat killed S strain plus R strain, live R strain, the second experiment, and yes, third experiment. Fourth experiment is nothing but it is a combination mixture. Both this two things are used here, heat killed and this one, both are mixed. Okay, I have written here again. So this is the mixture he was used. This mixture contained one heat killed S strain plus R strain live. Okay, he is not died the kill the R strain, no need to kill here. Okay, so both are mixed and injected to mice. When he injected this mixture of this heat killed S strain and R strain, what happens here? Mice is died. Mice died. Okay, so why it so happen? Okay, so this was killed. Yes, this was live. So that means something is transferred. Something is transferred from genetic material, that is transferred this virulent nature of the S strain into the R strain. Okay, so this is what the conclusion we can say. Let us we will write and see the conclusion. Okay. Yes, students, see here the conclusion of these experiments by Griffith. He concluded that the mixture when he injected here, mice is died, but alone when he injected, alone means here the single strain when he injected by killing, so that was not died. Okay, the conditions during R strain and heat killed strain, so during that mice is alive. Understood? Mice is alive. The same which he mixed and injected to the mice, so mice is died. So by these experiments his conclusion is, and also he was extract the live S strain. live S strain. Again he is extracted S strain, live S strain from mice, died mice. From this mice he was used the S strain. He was extracted, okay, he was extracted from dead mice. He was extracted the S strain. He observed that S strain live, which is live. He observed that, but he was injected dead S strain, but when injected into the mice, so that was become live. How it can happen? Because this S strain here, what he explained in his conclusion, he concluded that S strain bacteria, that S strain Streptococcus pneumonia bacteria, that was a virulent bacteria, had been transformed by heat killed S strain bacteria. R strain is transformed. The R strain is transformed by S strain, heat killed S strain, which is transformed. Okay. So this is what we call a transforming principle. According to this, so heat killed S strain had enabled, the R strain to synthesize the, one of the important thing here, smooth polysaccharide coat on that bacteria. Understood? So that is become virulent, hence it is able to kill that bacteria. Understood? So this heat killed S strain can enable this into virulent. So it was non-virulent. When it is alone, it was not virulent, but when it is mixed along with the heat killed, so it is become virulent. This is what the transformed. That genetic material is transformed from S strain to the R strain. That is what it is converted into a pathogenic form, virulent. So that R strain was not virulent when it is alone injected. When it is mixed with the heat killed S strain, it is become virulent, because of that can able to synthesize the polysaccharide coat on that bacteria. That is R strain were able to synthesize its polysaccharide, because that was the nature of S strain. Okay. So then it's become virulent, and this is what we call the due to transfer of genetic material, he explained, but he is unable to explain the biochemical nature, either it was done by RNA, DNA or any enzyme or protein, whatever. Okay. He was not explained regarding the molecular level of genetic material, what we call, but biomolecule nature, what we call biochemical nature, which is that biomolecule, he was not explained. That is what biochemical nature of the genetic material is remains unexplained by this experiment, series of experiments. Next, biochemical nature was explained by other scientists. Let us we discuss that in the next video. Subscribe the channel and please note the points. Okay. So this is one of the important for 5 marks. Okay. You can practice. Okay. Thank you, students.